Okay, I don't have any official disclaimers other than I did spend the afternoon on the beach and I had a beer, so that might affect my talk tonight. So, okay. So I am taking a little bit different angle, and you might argue that this is a little bit yesterday and old news, but I think there's a lot of parallels in terms of genomic risk profiling and how that just was adopted very rapidly by commercial entities, and that same thing might be seen as well with whole genome or whole exome sequencing. So I had in the title of the talk limitations. I took out the limitations, and really I should have put genomic risk profiling for prediction of common diseases, a skeptic's view. So we've heard a little bit about the promise of genomic medicine, and even before we were really thinking about clinical application of whole exome sequencing, we were getting very excited about finding sequencing through the Human Genome Project, and then finding various SNPs and so forth, and thinking that, wow, we're in the age of genomic medicine, and we're going to be able to do all these wonderful things by looking at everybody's genetic information. So really, basically saying we're going to take medicine from a one-size-fits-all approach to tailored care. Everybody's going to have different care, different medicines, different screening based on their genetic information and genetic makeup. And so we have several companies that thought this was a really great idea to use this information and be able to offer that genomic medicine direct to consumers. We have the ability to test someone's genetic risk for certain diseases and then make clinical decisions based on that genetic backdrop. Interestingly, several of the original companies have, like some of the whole exome companies, gone by the wayside, but the three that I'll mention here today are still in existence. However, very interestingly, with the exception of 23andMe, they are no longer direct-to-consumer. So now you have to involve your physician in ordering it, so that's really great. It's going to help the whole problem, as we'll see. So here's 23andMe. 23andMe, you can still, get on your own. And Navigenics, which has taken a little bit more of a upper-level road involving physicians. They tell us that we can incorporate your family history as well as your behavioral risk factors and provide you this healthier future. So where did all this come from? Well, it came from GWAS. So again, this seems old now, but it was pretty exciting at the time, where we figured out that we have these variations across the human genome, and we can use epidemiological principles to see which ones are associated with disease. So everybody knows what a SNP is, but just remembering that what we're doing here when we're doing genomic profiling is we're looking at a few 100,000 single nucleotide polymorphisms across the genome, not all 3 billion base pairs. And these are common polymorphisms, so at least 5% or greater in the population, and we know where they are, and there's a major and minor allele. And then what we do is basically use, again, epidemiological case control studies to figure out which of these SNPs are associated with a particular disease or trait. So you start with people with the disease, find similar people, at least you're supposed to find similar people without the disease, and then see if any differences pop up in the SNPs across the genome. Are they more or less likely to be found in your cases versus your controls? And so like classic case control studies, you end up with an odds ratio. And not to get into a bunch of epidemiology, but odds ratios were actually never meant to provide individualized risk prediction. They are a population-based measure. So lots of problems with the early GWAS. We were not accounting for multiple test effects, et cetera, and so forth, but we got better, and now this is the most recent off the NHGRI website. We have 249 traits and 1,600 plus verified SNPs associated with traits or diseases. So how do we use this information to predict risk? So we're looking at utilizing SNP-based information in healthy individuals to decide what we should do in terms of treating them different than the person down the road. So there's questions we have to ask. So are the SNP disease associations themselves valid? So are we using the right SNPs? Do we have a test that can actually pick up those SNPs in a good way? And then how do we use those SNPs to make the risk prediction? How many SNPs do we use? How would we combine those SNPs? Do we combine those with other non-genetic or non-DNA-based risk factors? And then how good are those risk predictions? So can they really tell me who's going to get the disease or who's not? And then if they can tell me who's going to get the disease and who's not, can I use that information in some way to change outcome? So is the GWAS valid? So this part we've gotten pretty good at. As I said, we deal with the multiple issues by increasing the requirements, stringency of our p-value. One of the problems has been disease phenotype variation. So when you're looking at these, you have to have lots and lots of cases and lots of controls. So you have lots and lots of cases. So you're looking at breast cancer. But not all breast cancer is the same. There's ER-positive breast cancer, ER-negative breast cancer, et cetera, et cetera, et cetera. But we're assuming that all breast cancer is the same. So it's difficult because just the description of that particular disease or how you're defining it is going to influence the validity of your SNP association or the strength of it as well. You have to deal with population stratification, and then can you replicate it? So people get really excited when they see p to the minus, 0.000008, right? But what we need to keep in mind is that just because it's highly statistically significant doesn't mean that it's a strong association. So it might be a highly significant association, but that doesn't mean it's a strong association. And really, if it's not a strong association, then we are unlikely to have much in the way of clinical significance. So for the vast majority of GWAS studies that have clearly found an association between a disease and a SNP, the odds ratios, in other words, the likelihood of disease in those that have the SNPs versus those that don't have that particular allele of interest, are really quite low. So typically in the range of 1.1 to 1.4 for the vast majority. So when we're dealing with odds ratios, that's not going to give us much in the way of individualized risk prediction. If we're looking at a population and saying, okay, this particular risk factor increases the likelihood of a disease in the population by 20%, we may say, okay, well, that might be something we want to pay attention to on a population level in terms of intervention, but is it really going to impact an individual in terms of their risk and medical management? So just an example, so we have an odds ratio with a particular SNP, and I'm just using SNP instead of the major or minor allele, but, and you see here some of the ways in which this is relayed to individuals or patients is you have an odds ratio of 1.3, that's a 30-fold increase. Well, I can tell you from a counseling perspective, if you tell somebody that they have a 30% increase, they're going to think a lot of different things, such as I have a 30% risk, et cetera, and so forth. But if you look at a relatively common disease even, so that affects maybe 1 in 100 individuals in the population, and then you say, okay, you have this SNP, so you're at 30% greater risk, so now your risk goes from 1% to 1.3%. So I can tell you that if you don't have this particular SNP, you have a 99% chance of not getting the disease, but if you have the SNP, you have a 98.7% chance of not getting the disease. Okay, so is that really helping us on an individual level? So we're pretty good at the analytic validity part, so I'll just rush right through that piece. The part that we're still lacking in and probably always will be lacking in, and I'll make a case for that, is the risk prediction part. So there's different ways to look at this calibration. Are the predicted risks in agreement with the observed? Is it generalizable? And can we take a predictive model that we created in one population, apply it to another population, and have it also predict well in that population? So clinical validity is actually asking the question, is it a worthy test? Is it a worthy test to do? So we look at sensitivity, specificity, risk distribution, which I'll illustrate a little bit, and then discriminatory accuracy. So when is a SNP useful? So it doesn't mean that odds ratios aren't important, as I illustrated earlier, and they actually, several of the GWAS have helped us find important disease loci. But even when you have a strong association, as I said, it doesn't mean that you have high sensitivity and specificity in terms of using that in a predictive fashion. The other thing is, when you have several SNPs that predict a particular disease, we pretty much will have all of us a distribution. So we'll carry a mixture of protective alleles and a mixture of risk alleles. So we all are pretty much ending up in the average range. There's only going to be a very small percentage of the population that are on the tail end of those distributions and have all of the high risk alleles or all of the low risk alleles. And it's really only at the tail end of those distributions that you can actually get some potential predictive risk information when you're dealing with low odds ratios. And again, I'll go into predicting for common disease, but also, is this giving us anything that we don't already know? I mean, can we get more than just we can get by taking a good family history, okay? So this is one of the arguments for why it's probably never going to have high predictive ability when we're dealing with common diseases, because we're dealing with genetic complexity. So we're dealing with most common diseases having many, many genes involved, many genes interacting with environmental factors. And the heritability tends to be much lower for these complex diseases than your monogenic disorders. And so by default, you're always going to end up in this sort of poor predictive because it's complex, you have low heritability. So why do we need risk prediction? Well, other than just for fun, we're supposedly using it, right, to make medical decisions. So how do we make medical decisions? Well, you essentially have to choose a cutoff. I'm either going to provide this intervention or I'm not, and I have to decide when that cut point is to provide that intervention. So on the upper part, you have an illustration of risk distribution for single gene high penetrance disorders. Well, you don't really have much of a distribution there, right? You basically have two peaks. Down below is illustrating a SNP profile for age-related macular degeneration. And I will say that's probably one of the handful of 2 or 3 conditions for which we can actually get some decent risk prediction with SNP-based profiling. But what you see here is you see not nice, you know, like this in terms of separation of your distribution curves, but at least you can kind of see where you might draw that line between treating and not treating, although you're still going to have, as you see, some false positives and some false negatives wherever you draw that line. But contrast that to looking at multiple SNPs for type 2 diabetes. And so again, the black are your patients with the disease, the gray are your controls, and you almost see complete overlap of distribution. I mean, where you put that cutoff, right? Where do you draw the line? And it's a measure of sensitivity versus specificity, but because there's such great overlap, you're not really going to have any decent predictive value. In either direction, you're always going to have a lot of false positives and a lot of false negatives. So the distribution is really what leads to the measure of discriminative accuracy, predictive ability. We usually use the measure of the receiver operator characteristic area under the curve, or often just called AUC. So what you're looking at here is a balance between sensitivity and specificity. You're assuming that those are of equal value, and we know that's not always the case. You might want to maximize sensitivity over specificity in some circumstances. But basically, if you have a test that's no better predictive than flipping a coin, then your AUC is 0.5, okay? So the higher the area under the curve, the better you are at predicting who will and will not get that disease. So here we have, again, a 10 risk loci profile for diabetes. These are all highly statistically significant, validated, replicated SNPs. And what we see is they give us an ROC of 0.57. Now remember, 0.5 is no better than flipping a coin, right? What about added value beyond traditional risk factors? So here we have the ACRS risk score, and looking at the clinical risk factors, we get that's not a bad AUC actually, but 0.76. So let's do genomic risk profiling and look at a bunch of SNPs on top of that, so we can really improve that AUC. Okay. And I would argue this can actually be improved because they don't do very well looking at family history with most cardiovascular risk scores. Okay. So this slide I kept in, even though some of it's older. I don't know if any of you remember this, but I think this was one of the first sort of ones to hit the fan, is when they found these 5 variants associated. And then literally the next day, there was New York Times saying for $300 you can get a risk profile for prostate cancer. So look at the AUC. So we can, with an accuracy of about 0.6, using age, geographic region, family history, predict whether or not someone will or not get prostate cancer. We can increase that by 0.02 if we add 5 SNPs to it. But if we just look at PSA and family history, we can get up to 0.92. Okay. So what's the problem here? Well, we don't know most of the disease-causing factors. We haven't identified all of the predictors or the genetic associations for common complex diseases. And what we've seen is that most of the identified SNPs account for only a small fraction of the known heritability of the disease. Now remember, when you're doing GWAS, you're looking at variants that are common in the population, okay? So we're not going to be looking at things that are less than 5% of the population. So it's quite possible we have all these much less common variants out there, but you're never going to find them with GWAS because of the way that those case control studies are done. So here you see why age-related macular degeneration is so good. We explain about 50%, and I think that's gone up. There's a recent article showing the impact of SNP profiling for age-related macular degeneration. We can do something with Crohn's disease, and then you can see it going down from there. This is just illustrating, again, some of the issues. So you know, this is what GWAS is finding, and the issue is we can keep finding more and more SNPs, but we're going to be finding more and more SNPs for common diseases that have odds ratios of 1.1, 1.2, 1.1, 1.05, or 0.98 if it's a protective allele, et cetera. So it's like, you know, what I remember learning with integrals. You keep inching, right? But you never get there. So one prediction is if we just kept going and we found 400 more undiscovered loci with allele frequency that you see there, odds ratio of 1.1. So if we found enough of them, then what we actually could end up doing is completely changing people's risk, who we said originally were threefold the general population, are now 0.3 the general population risk. So we're not going to find these rare variants. We quite possibly will find these rare variants with whole exome, but again, the question being how, even if we find a bunch of these rare variants, what is the strength of their association with the disease? So many of these companies are, they have marketing down to a T. They have patient education down really well. They have the use of the internet, et cetera. And so they will update you. Every time there is a new SNP that comes out that's validated, they will then add that to your profile and send you a new risk prediction. So how helpful is it? So here I am in year 1. And I'm told I'm at greater risk. So I have a predicted risk that's above the general population of 20%. And then they find 5 more SNPs, and they say, oh, okay, all 5 SNPs were now protective, so you're okay, don't worry about it. But then the year after that, they find 5 more SNPs, and you're back up into a high-risk category, okay? And that's because for most of these, again, we only know maybe 10% of the heritability. So as you keep adding to that, you can completely change someone's risk profile, theoretically. Okay, so is it a worthy test? So then is it worth testing? So you have to balance then the benefits versus the harms. Will using that test improve health outcomes? Are there interventions? Will it change medical management? And I'll talk a little bit about reclassification. The big thing now that everybody's using to defend genomic risk profiling is that people can choose then to do healthy behaviors. They'll be more motivated to jog or not eat those potato chips or what have you if they get that genomic risk profile back showing they're at increased risk for diabetes. Okay, so they change their health behaviors, but do they change them to an extent that it's actually going to do some good? Does it actually reduce morbidity and mortality? Again, these are all targeted towards the individual. Is there usefulness on a population level? Well, some people argue there might be. If we use SNP profiling to say, okay, instead of all women getting a mammogram at 40, we're going to stratify based on this and women in this category. So you're lumping again, but it's a population perspective versus an individual perspective. And then again, if there are benefits, are they worth the cost? How much does it cost the individual? Although, you get a sale with 23andMe, it's $100, so it's not too bad. What are the psychosocial implications? And then the thing that I'll point out again in a minute, but just because the test itself might not cost much, what if that test then leads to multiple physician visits or requests for additional tests or interventions and so forth that were potentially completely unnecessary? Given that the risk prediction is probably not terribly great. So one of the big arguments that some of the proponents of SNP-based risk profiling say, well, it's not all about the prediction of risk, it's about reclassifying people from one category into another. And if you can meaningfully reclassify people, then you can make different decisions. So what you're doing is, again, comparing these models based on traditional factors versus those based on traditional risk factors and genetic variants. And if you change them, then that's telling us that risk prediction model is useful. The difficulty is when they've looked at this carefully, although you might say, well, we did this test and of our population, we reclassified 6% of the population. So those 6% got the intervention that they should have when we would have missed them before. But the problem is if your predictive ability, if your area under the curve isn't good, you're just as likely to reclassify them the wrong way as you are to reclassify them the right way. And then the other thing is because, again, the amount of change in risk is so low, most of the people that you're reclassifying are sort of on the edge anyway. And for many of these diseases, we cut arbitrary lines. What's your hemoglobin A1C, right? We pick an arbitrary cutoff. And so it may be that we have to have odds ratios of 200 or more before actually reclassification would provide us with any meaningful information. Okay, so I'll just kind of wrap up with a big jab at Navigenics. I guess I picked on them because I kind of like some of the things I saw to pick on. So you're one of a kind, your health should be too, and they have all these wonderful, amazing success stories that are very exciting, very motivated. It's the end of illness. I just want you to know, if you do your genomic risk profile, it's the end of illness. So Navigenics, and I just blew this up. So Navigenics, they tell you, they offer you a more complete picture of your health by combining your lifestyle, health, and family history with your personal genetic information. Now, what that means to me is they are going to have a very sophisticated algorithm that takes all of the family history information and your behavioral lifestyle issues and your SNP, and they have prospectively validated this in a cohort of 250,000 people and shown that they can combine all those things and have a high predictive value. Well, I guess that's not what they do. They actually talk a lot about family history, but it's mostly, well, even if it's not in your family history, if you have this risk, it might be that. And I think they do have consultants, they do have genetic counselors that talk to people about, oh, well, you have this, so think about these health factors and so forth. But they are not actually truly looking at a model that incorporates all of those in any kind of predictive fashion. And this quote I love too: if your family health history seems relatively complete, okay, how many clinicians take a complete family history? I just want to ask that, other than those of us in this room. But your personal genetic test results will reveal exactly what you inherited. This isn't misleading, advertise it, is it? No? Okay. But it's okay, because we can click on the Find a Physician link, and we can find the physician that will help us understand these results and will help us order them, because now I can't order them anymore DTC because of the whole GAO report, and got some bad press about that. So I thought, okay, I'm gonna look for these physicians. So I clicked on Georgia, and a physician came up in Atlanta at Emory. He's an internist, and he's certified in densitometry. And when I looked at his profile, I really didn't see anything to do with genetics whatsoever in his profile. Okay. So you can find a physician that will help you order that test, that will help you interpret that test. Is it an appropriate physician? So this is looking at an example of how they provide this risk profile information. First of all, any of you have ever worked with patients and done genetic counseling? This is really kind of, wow, how do I deal with all this information and these numbers? And then, of course, you get really excited about everything that's in orange, right? Because that's the scary stuff. So here I am, my heart attack is in orange. But wait, my risk is this average of the general population. But they put everything in orange that's a 25% risk or greater, okay? And then obesity, it's orange, okay? But my risk is actually 7% lower than the general population, but it's still 25%, so I'm still in a high-risk category. Okay. The other thing about this, and you probably have heard this, but there have been several studies where they've had individuals submit blood samples to several different labs and get their profiles back, and there's I think it's around 30% discordant in results. So one lab will say you're at higher risk for breast cancer, and another lab will say you're at lower risk. They use different SNPs, they use different risk models, and they use different background population figures. So these are odds ratios, so they generate your individual risk by basically multiplying the population risk by your odds ratio. So, it's just fun, right? People can just do it, it's no big deal. So what's the problem? And there's been a couple studies saying that people are not being psychologically damaged by this kind of profiling, and people are suggesting that maybe even be doing positive health behaviors. Well, at least they say they're going to be doing they say it's going to motivate them to have positive health behaviors. Well, those of you who've been in public health know that what people say they're going to do isn't often what they're going to do. So But what if the risk prediction is inaccurate? What if we provide them false reassurance? So you have a woman who has a family history of breast cancer. She's really worried and she wants to know she's at genetic risk, so she goes and does a 23andMe, and she's come back and she's reassured because her risk for breast cancer is less than the general population, but lo and behold, there's a BRCA1 mutation in her family. And then by the flip side, people have the orange box, and they're all freaked out because they have a 32% instead of a 31% risk for diabetes. And they ask for all these tests, and the doctors tend to give them to them. And this is just an example of what we've seen. Patients think their doctors can explain it, and they expect their doctors to do something about it. That most people will share, and they believe the results implies a medical diagnosis. So if I'm in that orange box, that means I have diabetes. And yet, of primary care providers, most don't think they have the ability to answer those questions. And I found this one particularly ironic. So this is a group of providers who are, I think, in that pool that sign up for offering tests for Navigenics. So they've ordered the test for themselves or others, but 60% believe there's no utility to it. I think that goes against the Hippocratic Oath, I'm not sure. Anyway, so the bottom line is predicting complex disease is complex. And no matter how many variants we find, you can't actually develop an individualized risk profile because everybody is unique. So if I have 5,000 variants that predict a certain disease, that means I have to find 5,000 other individuals with that exact same profile, or many thousands with that exact same profile, which is just the more variants you add, the less likely you're going to find enough people with that exact profile to be able to compare their disease risks. So I'll just end with that and say, we've come a long way, baby, but I don't think we're quite all the way there yet. Thank you.